So, as part of this, aren't too many other faculty colleagues. And I thought, well, I should have a faculty colleague list in here. And I mentioned Jim. He was a tremendous associate for a long time. But Jeffrey is really the one who steered me on the right course to begin with. And Jeffrey Granett is here. He's an entomologist for a goodly number of years. And he's going to tell you about his interactions with me, I guess, and searching for Phylloxera and where we did such. And it's been a nice, long relationship. It's been great. Some of the things you don't know about Jeffrey, if you know him at all, probably, is that he's an amazing artist, a really amazing artist. And his woodblocks are my favorites. So if you've ever been at my house, you've barely commented about this strange woodblock I have in the bathroom. You're thinking, this guy is very strange. There's a strange woodblock in here. And that's Jeffrey's woodblock. It's really a spectacular piece of art. So
I'll show you one woodblock on my show today.
Perfect, perfect.
Okay, good afternoon. Thanks for the introduction. The title of my talk is Walking the Creepy Crawlers. My insect, grape phylloxera, is tiny and can walk around a lot when challenged. Hence, it is a creepy crawler. And thus comes the title. Each creepy crawler is a walker. I retired from the entomology department at UC Davis about 15 years ago and have not been formally involved in science or research since. Prior to retirement, my interactions with Andy were on Phylloxera research and outreach of the research results to grape growers. Today I will give you a very brief overview of the insect's interaction with grapes because that is what Andy did. Yes. My research with grape phylloxera involved lab and field work and began about 1983. This insect can live on grape roots by swelling the root cells, which increases the nutrients floating around in the cell and that it feeds on. You can see the adults and lots of eggs in this picture. Do you know which are which? The yellow ones are the eggs, and the larger ones are the adults. The next picture shows the galls on a non-vinifera grape leaf. Each gall bump on the leaf has enclosed space inside for a single adult phylloxera, which will then lay many eggs. When these eggs hatch, the young insect will leave the gall and move to another leaf and make a new gall which spreads the infestation. Although phylloxera are so small that they are individually almost invisible without a magnifying glass or the root swellings and the leaf galls they cause on the vines are large enough to easily see them when you visit an infested vineyard. The life cycle is easy to follow. So, do you know which one's the adult? The one with the wings. And down below here, there's another adult that doesn't have wings. So they either develop wings or they don't. Depends on where they are in the situation. So they lay eggs. The eggs hatch into nymphs. The nymphs molt 4 times to become adults, and the cycle starts over again. Some phylloxera feed only on roots. So you can see the infested roots in this illustration. It's bumpy and black and looks horrible, unless you're a phylloxera. Where was I? On occasion, they form wings and spread the infestation that way to previously uninfested vines. The young ones, as well as the winged ones, can blow in the wind and spread that way as well. Andy and I have about 20 multi-authored scientific journal publications on this insect. I decide not to read them here. With my insect skills, I could set up lab or field experiments in which I could watch the insects appear, grow, and increase in numbers. But Andy's expertise and skills were critical for determining the impact of cultivars, weather, and vine conditions. This picture is from a set of infrared photos of a Napa Valley vineyard that Andy found for us to work with. The aerial view told us where to look for populations when we went to sample. The red rows are the vines that are relatively healthy, though they may be infested. The damaged or dead vines are the tan or dark spots between. In some of these blocks, there's a lot, and some of them, there's just a few. When sampling, we frequently found the insect at the edges between healthy and damaged vines. So in this picture, there are healthy vines, which obviously are green, though they may be infested, and the dead vines were killed off by Phylloxera populations on the roots. Okay. In Southern California, in the backcountry, we found that the indigenous ancient peoples literature on the rocks told us where to sample. Translation of the text seen in this slide required a lot of dictionary work on the part of Andy. Would you like to read it now? Sampling isn't just a casual walk through a vineyard. It requires serious digging, lots of hard labor. We did a lot of field sampling, surveying, and tests. Experiments sometimes required a crew with shovels, and sometimes shovels were just not sufficient. With his viticultural knowledge and connections, Andy could find test vineyards with rootstock cultivars. Similarly, for lab tests, he could find the right plant materials. Our research identified how different rootstock cultivars and growing conditions were associated with insect biotypes, variation, and movement. The work suggested that cultivars are either supportive of Phylloxera strains or not. We learned the limitations of insect biotypes and how to avoid problems in vineyards with them. We studied Phylloxera biotypes through wild as well as cultivated grape areas. Being part of the agricultural experiment station, it was our responsibility to convey our results to those who managed vineyards. This is a picture of Andy at an extension outreach seminar. Andy is great in dealing with growers. He always knows what he's talking about with them. He knows their problems and how they might go about solving them, and he has a really good sense of humor. Of course, as you know, Andy is a fine speaker and gave many, many talks during his long career, and his work efforts have always been well focused. His move to retirement will require more than a new set of tires for his car. That's the retiring with the dash. Andy will have a new organization to his life without a formal job or much of a formal job. He will need to keep his mind and muscles active, have the energy to run after the grandkids, be creative with vacant morning coffee break time, and cultivate those orchids, and of course enjoy life and the occasional sip of wine. Andy, although my science might not be up to date, my retirement skills developed well and will develop well for you as well, I am sure. Good wishes and best of fortune to both you and your family.
By the way, if you want a close-up view of the Phylloxera shirt, We have them for sale back with our CDs in the back there.